Hi, friends. Today's topic is DNA vaccine, synthetic peptide vaccine, and multivalent vaccine. So, what is a vaccine? It is a substance used to stimulate the production of antibodies and provide immunity against one or several diseases, and it is prepared from the causative agent of a disease or its products or a synthetic substitute, and is treated to act as an antigen without inducing the disease. It helps your immune system recognize and destroy the living microbe during a future infection. Therefore, process vaccination is the administration of vaccine to help immune system develop protection from disease. DNA vaccine. DNA vaccine involves a genetically engineered plasmid which contains a particular DNA sequence which can encode antigen against which an immune response is needed. This plasmid is injected into the appropriate tissue. It offers both B and T cell responses and improved vaccine stability and also the absence of any infectious agent. The advantages of DNA vaccines over conventional vaccines include the ability to induce a wider range of response types. The viral antigen is expressed not only by the muscle cells but also by dendritic cells in the area that take up the plasmid DNA and express the viral antigen. Plasmid DNA encoding the antigenic proteins is injected directly into the muscle of the recipient. The muscle cells takes up the DNA and encoded protein antigen is expressed, which leads to both humoral antibody response and a cell-mediated response. Injected DNA is taken up and expressed by the muscle cells with a much greater efficiency than in tissue culture. Advantages. The advantages over many of the existing vaccines include that the encoded protein is expressed in the host in its natural form and therefore no denaturation or modification is done. The immune response is directed to the antigen exactly as it is expressed by the pathogen. It can induce both humoral and cell mediated immunity. DNA vaccines can also cause prolonged expression of the antigen, which generates significant immunological memory. Refrigeration is not required for the handling and storage of the plasmid DNA, and this greatly lowers the cost and complexity of delivery. The same plasmid vector can be custom tailored to make a variety of proteins so that the same manufacturing techniques can be used for different DNA vaccines, each encoding an antigen from a different pathogen. The development and production is easy, and the storage and shipping are stable and cost effective. Method for administering these vaccines entails coating microscopic gold beads with the plasmid DNA and delivered the coated particles through skin into the underlying muscles with a gene gun. This will allow rapid delivery of a vaccine to large populations without the requirement for huge supplies of needles and syringes. Here in this figure, we can see that the plasmid vaccine, when introduced, induces both cellular and humoral immune response. When the gene is inserted or injected into the muscle cell, it gets expressed in the injected muscle cell and in the nearby APCs or otherwise antigen presenting cells. So, here we can see that the peptides from the protein encoded by the DNA are expressed on the surface of both the cell types after process. Cells that present the antigen in the context of class 1 MHC molecules stimulate the development of cytotoxic T cells. Like here we can see that's the balance of the figure. It is in the next slide. So here we can see the development of cytotoxic T cells, and in the next pathway the protein encoded by the injected DNA is also expressed as a soluble secreted protein which is taken up, processed and presented in the context of class 2 MHC molecules. This pathway stimulates the B cell immunity and generates antibodies and B cell memory against the protein. Disadvantages. This includes the limited to protein immunogens. It is limited to protein immunogens, that is, it is not useful for non-protein based antigens like bacterial polysaccharides. It may affect the genes which control the cell growth. There is also a possibility of inducing antibody production against the DNA. And also, tolerance may be developed to the antigen produced. Potential for atypical processing of bacterial and parasite proteins can be seen. Tests of DNA vaccines in animal models shows that these vaccines are able to induce protective immunity against a number of pathogens, including the influenza virus. Inclusion of certain DNA sequences in the vector may lead to enhanced immune response. At present, there are human trials underway with several different DNA vaccines, including those for malaria, AIDS, influenza, and herpes virus. Let's discuss the synthetic peptide vaccine in the next part of the video. Thank you.